Let's continue with our topic of organelles and cell structures. So this is part 2. So in part 2, we're going to talk about the ribosome. The ribosome is a structure found inside cells that is found in all cells, in prokaryotes, bacteria, and eukaryotes, everything else. What does a ribosome do? It makes proteins. Fancy word for make, synthesize. As you will see, as you continue to see, there's a lot of higher vocabulary that we use in biology. So have to get used to some of these higher vocabulary words like synthesize. Now, the ribosome does not have a membrane around it. So that means it's not a membrane-bound organelle, which is the definition of difference between eukaryotes and prokaryotes. And that is why we find it in both. It's simply made out of rRNA. RNA is a nucleic acid we talked about, nucleotides strung together. The R in the front just tells you that it's a part of a ribosome, so it's called ribosomal RNA. And proteins. So it's made by the nucleolus, which we already talked about. It has 2 parts. It has a large subunit, a small subunit. They hook together, and right where they meet is where the mRNA binds, and then is read to make the protein. Ribosomes are either attached to an ER, which is the endoplasmic reticulum, or they're floating free in the cytoplasm. So what is an ER? Endoplasmic reticulum, ER. In general, its job is to transport and move materials around the cell. So when you think about the cytoskeleton, its job would be to move organelles around the cell. The endoplasmic reticulum moves everything else. And it's made of something called cisternae, which only tells you that it's a bunch of membranes, but instead of being a little round sphere like the next few organelles look like, it either looks like tubes or it looks like these round sacs. So just cisternae is just the name of these shapes here. So there are 2 kinds of ER. The first one is called rough ER, or the rough endoplasmic reticulum, or the RER. Really easy when you say RER. And what does it do? It produces, and it transports membranes. So remember what membranes are. They are phospholipids. So very specifically, in the RER, it makes membranes. And it also produces and transports proteins. Proteins that are going to be secreted or released. And that happens because the ribosomes make proteins and they're attached to the RER. So this is the structure. The structure is that the cisternae, and they're covered in ribosomes. I'm assuming that people probably thought if you were to actually be able to touch this ER and you touched it, you'd feel all these bumps, the ribosomes, and so it would feel rough. And I think that's where they got the name. It looks rough. So once produced, proteins, they pinch off in something called a vesicle. It's just a sac. It's just a little circle, a little 3D sphere. It always is a membrane. And the protein's on the inside. And then that vesicle travels to the Golgi for processing. We'll talk about the Golgi in a second here. This is a micrograph to show you what it would look like in real life without drawings. So in real life without drawings, this part is the RER, the membrane part here. And then the dots that are touching the membrane are called bound ribosomes to the RER, to the ER. And then the dots that are not touching are free, right? So free ribosomes, just hanging out. Then we also find smooth endoplasmic reticulum, smooth ER, SER, whatever you want to call it. Its job is to make and transport lipids. So wait, back here, membranes, phospholipids. Here, lipids only. So not the membranes, but those other kind of lipids like oil and wax and other things, right? That we talked about, triglycerides. In muscle cells, we actually have quite a bit of smooth RER because it stores and releases the calcium ions in our muscles. And we use calcium ions for our muscles to contract. So not only do we need calcium for our bones, but we also need calcium for our muscles to work correctly. And our smooth endoplasmic reticulum is going to take care of that. And then we also have enzymes in the SER of liver cells that are going to help process drugs and other potentially harmful substances. So if you had a poison or any sort of other drug, your SER is in charge of helping to break that chemical down and to clean out our blood. That's what your liver's job is. So your liver cells have some SER in them. And the big thing about the structure is that they have no ribosomes. So they're smooth. I guess if you were to touch them, they would be smooth. Not that you could. So here's a picture, a real life picture of what it looks like. And you can see the little membranes, and they're just smooth, right? Because compared over here that have all these dots attached to them, that's the rough. So the rough has all the dots, all the ribosomes. The smooth, no ribosomes. Okay, the next part of organelles that we have to learn is the Golgi. Your book calls it the Golgi apparatus. I think I learned it as Golgi body. It seems to change a lot. Depends upon which book you read and the author. So just look for the word Golgi and you'll be safe. What does the Golgi do? They always call it the post office. Like it modifies and packages the protein. So that's what a lot of people call it, the little post office. So everything that comes out of the ER, either the smooth or the rough, is going to pinch off, create a vesicle, which is just a sphere of membrane with stuff inside. It goes to the Golgi. The Golgi like repackages it into a different vesicle, or at least put something on the outside like a label, and then it sends off a new vesicle with that thing inside. And then that thing inside either is going to stay inside the cell, like it's going to become a lysosome, or that vesicle is going to go to the plasma membrane, and whatever protein was made is going to get released from the cell. So it has various jobs there. And it's very flattened. So the structure is very flattened. See here? It looks like it's really flat. This is the Golgi. It's a little different looking than the endoplasmic reticulum. So again, just real flat structure here, the Golgi. All right, so the lysosome, we just mentioned that. When our ER makes proteins, it's either making proteins to be released by our cell, or it's making proteins called enzymes, which we already know about, right? Enzymes. So when the ER makes enzymes, when the RER makes enzymes, it's going to send that vesicle to the Golgi, and then the Golgi is going to relabel it as a lysosome, and then it will release it out. And a lysosome is a membrane-bound sac that has digestive enzymes on the inside. And what does that lysosome do with those digestive enzymes? Well, it helps digest food particles inside the cell. It also helps destroy bacteria, especially that's in our white blood cells. We have special lysosomes inside our white blood cells that destroy bacteria. It also removes or recycles damaged parts like organelles. So in this picture, the lysosome is going to recycle the mitochondria because the mitochondria is not working anymore. So it will digest it and split it apart into its pieces, and then maybe a new mitochondria can get made. Or this waste will just be released because we don't need it. So it kind of keeps everything all nice and clean inside the cell. It's a really good cleaner, recycler of stuff. So it keeps our cells functioning. So with the digestive enzymes, remember enzymes either break things apart or put things together. So these are probably ones that break things apart. They live inside the lysosome in a pH of 4.5, which is really acidic for a cell. So That's probably because it helps the digestive process. That means that these enzymes love living at 4.5 pH. All the other proteins that are out there probably do not like 4.5 pH, and that's why it works as a digestive system. Um, and then I just told you the Golgi is what like kind of creates the lysosome after the ER makes the digestive enzyme. All right, another kind of thing that looks like a lysosome, but very specialized lysosome is called a peroxisome. It neutralizes free radicals. A free radical would be like an oxygen ion. Oxygen loves to bond to stuff, and especially when it's an ion. Remember, it loves electrons. It wants electrons. So oxygen, when it's an oxygen ion and it's floating around our body inside our cells, it will bond to the chemicals in our cells and damage them. So those aren't good. So we have something to take care of that. They also break down fatty acids, which can also then go to the mitochondria to help make ATP. And they specifically detoxify alcohol. So your SER also helps detoxify alcohol and other drugs, but specifically the peroxisome, you might have extra peroxisomes for alcohol. When it does that to the alcohol, it actually makes hydrogen peroxide, which you are familiar with if you've ever seen the brown jar in your bathroom, right? That your mom puts on your cuts to clean it out. So that's actually how it gets its name, peroxisome, because it makes hydrogen peroxide when it breaks down alcohol or when it kills the bacteria. So it too is just a membrane-bound sphere. It looks just like a lysosome, but it has just a different job, so it has a different name. And that leads us to notes review number 3.